Hello, everyone, and welcome to today's live broadcast, High-Throughput Proteomic Applications Using Anti-DYKDDDDK Magnetic Agarose, presented by Dr. Betsy Benton, R&D Staff Scientist and Protein and Cell Analyst. I am Xavier Gutierrez of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots and sponsored by Thermo Fisher Scientific. For more information about our sponsor, visit www.thermofisher.com. Now, before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during this poster presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the poster presentation. If you have trouble seeing or hearing the poster presentation, please use the Ask a Question box to let us know that you are having a problem. This poster presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the poster presentation window and follow the process to obtain your credits. Now, please join me in welcoming Dr. Benton. I will turn the poster presentation over to her.
Thank you, and hello. I will be sharing with you several proteomic applications that we have performed using our anti-DYKDDDDK magnetic agarose and the Kingfisher Flex magnetic particle processor. The DYKDDDDK tag is a common epitope tag used for protein purification. The 8-amino acid tag is most often referred to as the flag tag, a name that was coined and trademarked by Sigma. In the interest of simplicity, I will refer to DYKDDDDK as flag throughout the remainder of the presentation. The flag tag is a highly versatile tag in that it can be inserted onto the N or the C terminus of proteins that are expressed in bacterial, mammalian, yeast, and insect cells, as well as in vitro expression systems. The flag tag is recognized by a high-affinity rat monoclonal antibody that is covalently attached to our high-capacity magnetic agarose. Throughout the course of this presentation, I will demonstrate effective automated purification of flag tag proteins from multiple complex biological systems, focusing on three key features that are most important to researchers: high yield, high purity, and biological activity. Table 1 shows the characteristics of the Pierce anti-flag magnetic agarose. The beads are composed of magnetic, highly cross-linked 6% agarose. They are ferromagnetic with low remanence, meaning that upon release from a magnet, the beads do not maintain significant magnetism and can be fully dispersed. The bead diameter is between 10 and 40 micron, with an average of about 25 micron. The beads are provided as a 25% slurry in PBS containing 0.01% tween-20, along with sodium azide as a preservative. The binding capacity is greater than or equal to 3.2 milligrams of flag-tagged green fluorescent protein, or GFP, per mil of settled beads. And lastly, the flag-tagged proteins can be captured in as little as 20 minutes. Figure 1 shows the purification of N and C-terminal flag-tagged SUMO protein that were overexpressed separately in E. coli. The proteins were captured with 12.5 microliters of settled anti-flag magnetic agarose, washed, and then eluted with 0.1 molar glycine with a pH of 2.8. The stain gel shows each lysate and the purified proteins in duplicate. Negligible nonspecific binding to the beads is resulted in high purity protein recoveries as shown. Figure 2 shows the purification of 1X and 3X flag tag TNF-alpha from XB-CHO media. Each protein was expressed separately in the XB-CHO expression system, where they were secreted into the media. Purification of the proteins was performed in replicates of 6 on the Kingfisher Flex instrument. 0.4 mils of clarified media was incubated with 50 microliters of settled beads for 1 hour. The beads were washed and then eluted with 0.1 molar glycine at pH 2.8. The load, flow-throughs, and elutions were evaluated by stained SDS page in panel A. The left side of the gel shows minimal to no protein remaining in the flow-throughs, indicating efficient capture. The right side of the gel shows single equivalent bands of eluted protein with no background. Protein yield was measured in each eluate by protein assay shown in panel B. The average amount of protein purified was about 110 micrograms of the 1X flag tag TNF-alpha and 162 micrograms of 3X flag tag TNF-alpha. The standard deviations were less than 3 and corresponding CVs were less than 3%. It's important to note that the difference in protein yield is not reflective of the difference in tag length, but rather in the difference in expression efficiency. Each purified protein was further evaluated using a BioCore instrument, and their binding kinetics are shown in panel C. Flag antibody was captured on a chip using a goat anti-rat IgG, and then the purified proteins were passed over the chip. The data shows a 4-fold faster on rate, or Ka, for the 3X tag, but the off rate is similar for both tags, allowing for a 20 to 60 minute bind or capture step. There is essentially no difference between the 1X and 3X flag tags at equilibrium. Figure 3 shows the activity of the purified 1X and 3X flag tag TNF-alpha. HeLa cells were treated with and without tag TNF-alpha, as well as TNF-alpha without a flag tag. Following 20 minutes of treatment, the cells were lysed and partitioned into cytosolic and nuclear fractions. The fractions were analyzed by Western blot for the nuclear transcription factor NF-kappa B P65, which is known to translocate from the cytosol into the nucleus upon cell treatment with TNF-alpha. The treatments are shown across the top of the blot, and cytosol and nuclear fractions are designated as C and N, respectively. In the absence of TNF-alpha, nearly all of the NF-kappa B is found in the cytosol. In contrast, TNF-alpha, both with and without a flag tag, is shown to promote transport of NF-kappa B into the nucleus. The data demonstrates that purification with anti-flag magnetic agarose maintains the biological activity of purified flag tag protein. Figure 4 shows the purification of C-terminal flag-tagged green ranilla luciferase expressed with a high-yield in vitro translation kit. The protein was immunoprecipitated with Pierce anti-flag magnetic agarose and Sigma's anti-flag M2 magnetic beads on the Kingfisher Flex. The beads were washed and then competitively eluted with 3X flag peptide. Panel A shows a Western blot of the starting lysate, flow-throughs, eluates, and post-elution bead boils. Results for duplicate samples are shown, and all fractions were normalized by volume. Significantly higher yield was seen with the Thermo Scientific anti-flag magnetic agarose. To evaluate nonspecific binding to the beads, the eluates were resolved on a gel and silver-stained. The only band visible is the flag-tagged green ranilla luciferase, indicating very low background with both thermo and competitor anti-flag magnetic beads. Panel C shows the relative fluorescence units of the eluted flag-tagged green Ranella luciferase. The amount of active protein seen with the anti-flag magnetic agarose is about 5 times higher than competitor beads, corresponding to the Western blot showing significantly more eluted protein with the Thermo Scientific beads. Overall, the data shows that eluted protein maintains biological activity throughout purification. Mass spectrometry is a key proteomic application that can be used to identify multiple protein-protein interactions in a co-immunoprecipitation. In Figure 5, C-terminal flag-tagged bad protein was transfected into HEC293 cells, and then bad protein was immunoprecipitated from cell lysate with anti-flag magnetic agarose on a Kingfisher flex. Non-transfected cells were used as a control. Panel A shows a Western blot of both lysate and eluted protein that were probed with anti-bad antibody on the top panel and anti-flag antibody on the bottom. Bad is clearly seen in both lysate and eluate of transfected cells, but not in the control. The eluates were next analyzed by mass spectrometry. To do this, the eluates were processed in solution, digested with trypsin, and analyzed by nano-LC-MS-MS using a Thermo Scientific Dionex nano-LC system and a Q-exactive HF mass spectrometer. The top 3 peptides identified for the eluate from the lysate with expressed flag tag bad corresponded to the bad protein. The non-transfected control did not contain any bad peptides. Next, protein-protein interactions were identified using Thermo Scientific Proteome Discoverer 2.2 software and string database. The bad protein, as well as multiple known interactors, including several isoforms of the protein 1433 and mitogen-activated protein kinase 9, were identified. Panel D shows the interactions of all of these proteins with bad in a string diagram. Figure 6 shows the immunoprecipitation of nanogram quantities of flag-tagged protein that was spiked into a mammalian cell lysate. Here, 25 nanograms of N-terminal flag-tagged green fluorescent protein, or GFP, was spiked into A549 cell lysate and captured using anti-flag magnetic agarose or Sigma's anti-flag M2 magnetic beads using the Kingfisher flex. Tagged protein was eluted with 0.1 molar glycine at pH 2.8 and analyzed by Western blot. Going from left to right, the starting lysate is shown, followed by duplicate elutions and duplicate post-elution bead boils. Yield of tagged GFP is seen to be higher with thermomagnetic agarose versus competitor beads, indicating that the higher capacity thermal beads are more effective at capturing low abundant targets. The eluates were also run on a silver stain gel to evaluate nonspecific binding. The only band visible is that of the tagged GFP with the Thermo Scientific beads. Background for both thermo and competitor beads is seen to be negligible. In conclusion, the Pierce anti-flag magnetic agarose purifies biologically active flag-tagged proteins with negligible nonspecific background. Both the 1X and 3X flag-tagged proteins are purified with similar efficiency. Mass spectrometry can be used to identify protein-protein interactions from immunoprecipitations and co-immunoprecipitations with anti-flag magnetic agarose. And lastly, both abundant and low abundant flag-tagged proteins, ranging from nanogram up to milligram quantities, can be efficiently purified from complex biological systems with Pierce anti-flag magnetic agarose. This concludes our presentation. Thank you for your attention. I'm happy to answer your questions.
Thank you, Dr. Benton, for that informative poster presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your poster presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many of your questions as we have time for. Now let's begin. Our first question is, what are the benefits of using high-capacity magnetic beads with co-IP?
High-capacity beads work very well for capturing low-abundant targets. You saw that in the last figure of the presentation, figure 6. The reason for this is that due to the higher capacity, you can capture more of the available target in the lysate. We've seen higher yield in both Western blots as well as in mass spectrometry of IP lysates. You can also use, in the case of magnetic beads, where say you have a higher capacity than you might need, you can use less beads, which will lower your cost per sample. And in addition, if you can use less beads, potentially you can reduce your background if you're working with a particularly sticky sample.
All right, and can the flag tag be removed?
Yes, the flag tag can most definitely be removed. Enterokinase is the enzyme that can be used to remove the flag tag on the N-terminus of a flag tag protein. The enzyme recognizes the full flag tag, the DYKD4K sequence, cleaving after that last lysine residue, completely removing the tag from the protein.
Okay, and looks like we have one more question. Where can I find Kingfisher protocols for the anti-flag magnetic agarose?
Our Kingfisher protocols for the beads can be found on the product page online at thermofisher.com. Specifically, if you're looking at the product page for the anti-flag magnetic agarose, you would go to the bottom of that page under product literature where the links are listed.
Well, I would like to once again thank Dr. Benton for her poster presentation. I would also like to thank Thermo Fisher Scientific for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through September of 2019. Please share with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us, and we hope to see you again soon. Goodbye.